Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The landscape of scientific inquiry often reveals its transformative potential through the refinement of existing methodologies and the introduction of revolutionary techniques. In the realm of biochemistry, the separation of complex mixtures of biomolecules presented a formidable challenge throughout the early 20th century. The ability to isolate and analyze individual components, such as amino acids and peptides, was crucial for unraveling the intricate workings of biological systems. Richard Laurence Millington Synge, in collaboration with Archer John Porter Martin, rose to meet this challenge, profoundly altering the course of biochemical analysis. Their pioneering work culminated in the development of partition chromatography, a technique that would revolutionize the field. The problem Synge and Martin addressed was fundamentally one of selectivity and efficiency. Existing methods for separating complex mixtures, such as fractional distillation, were often inadequate for the separation of complex biochemical compounds. These compounds, frequently possessing similar chemical properties, proved difficult to differentiate using traditional approaches. Furthermore, the fragility of many biomolecules demanded gentle separation methods that minimized degradation. The need for a versatile and reliable method for separating amino acids and peptides, essential building blocks of proteins, was particularly pressing. The ability to isolate and identify these components was a prerequisite for understanding protein structure and function, a burgeoning area of research at the time. Synge and Martin's solution was elegant in its simplicity and revolutionary in its impact. They devised partition chromatography, a method based on the differential partitioning of compounds between two immiscible liquid phases. This approach leveraged the differing solubilities of the target molecules, exploiting their varying affinities for the stationary and mobile phases. This technique contrasted with earlier methods that relied on differences in volatility or charge. Their method employed a liquid stationary phase, such as water, held on a solid support, typically silica gel, and a liquid mobile phase, such as an organic solvent. The mixture to be separated was introduced to the column, and the mobile phase was passed through it. As the mobile phase moved, the components of the mixture distributed themselves between the two liquid phases, separating based on their relative solubilities. Compounds with a greater affinity for the mobile phase would move through the column faster, while those with a stronger affinity for the stationary phase would move more slowly. The specific tool they created was the partition chromatography method itself, comprising the column, the stationary phase supported on a solid matrix, the mobile phase, and the means of introducing the sample and collecting the separated fractions. The choice of materials was critical. The stationary phase had to be carefully chosen to provide the desired selectivity, while the solid support had to be inert and capable of holding the stationary phase without interfering with the separation. The mobile phase had to be compatible with both the stationary phase and the compounds being separated. The careful selection of these components was an essential element of their innovation. A tangible example of their work can be found in their seminal 1941 paper, which detailed the successful separation of amino acids using partition chromatography. In this landmark publication, Synge and Martin demonstrated the ability to separate a mixture of amino acids, a feat that had previously proven extraordinarily challenging. They meticulously described the experimental setup, the materials used, and the conditions required to achieve effective separation. This work provided a proof of concept, demonstrating the power and versatility of their new method. The ability to separate amino acids opened up new avenues of research, allowed scientists to study protein composition, analyze the products of protein hydrolysis, and develop new methods for protein sequencing. The modern relevance of partition chromatography is undeniable. While the original method has undergone significant refinements and evolution, the fundamental principles remain at the heart of a vast array of chromatographic techniques used today. These techniques, including high-performance liquid chromatography, or HPLC, are indispensable tools in biochemistry, pharmaceutical analysis, environmental science, and numerous other fields. HPLC is used to separate and analyze a wide range of compounds, from small molecules to large biomolecules. The ability to separate complex mixtures is essential for quality control in pharmaceutical manufacturing, for identifying and quantifying pollutants in the environment, and for understanding the complex interactions within living organisms. The legacy of Synge and Martin's work continues to shape scientific progress. Synge's understanding of the principles of liquid-liquid extraction likely influenced his approach to partition chromatography. This technique, well established in the chemical industry, involved separating components of a mixture based on their differential solubility into immiscible liquids. Synge, with his background in physical chemistry, would have been familiar with these concepts. The work of chemists investigating solvent extraction processes provided a foundation for his thinking. The understanding of how solutes distribute themselves between two phases and the factors that influence this distribution was critical to the development of partition chromatography. The work of previous scientists in the field of solvent extraction directly contributed to the conceptual framework that Synge and Martin employed. Their work built upon existing knowledge, adapting and refining it to address the specific challenges of separating complex biochemical mixtures. The impact of Synge and Martin's work extended beyond the immediate application of partition chromatography. It laid the groundwork for the development of subsequent chromatographic techniques. Scientists in the years following their initial publication built upon their foundation, leading to the development of new and improved separation methods. Their work served as a catalyst, inspiring further innovation and refinement in the field. The principles they established continue to guide researchers in developing new separation technologies and understanding the fundamental processes that govern chromatographic separations. The influence of their work is seen in the evolution of chromatography, a field that continues to advance and provide indispensable tools for scientific discovery. The conceptual framework that Synge and Martin employed, as we have seen, was a pivotal moment in the history of separation science. Their work adapted and refined existing knowledge to address the challenges inherent in separating complex biochemical mixtures. This initial breakthrough, however, was merely the prelude to a more refined understanding of the underlying principles, a refinement which led to the development of partition chromatography. The intellectual problem that Synge and Martin confronted centered on the need for a method capable of separating complex mixtures mixtures of biochemical compounds. Traditional methods, while useful, often proved inadequate when dealing with the intricate compositions of biological samples. They sought a technique that could differentiate between compounds based on their differing affinities for two immiscible liquid phases. This approach, rooted in the established principles of liquid-liquid extraction, offered a novel solution. The core concept centered on differential partitioning, the differing solubilities of individual compounds within a mixture between two liquids that do not mix, a stationary phase and a mobile phase. This difference in solubility, driven by intermolecular forces such as van der Waals interactions and hydrogen bonding, would dictate how each compound would distribute itself between the two phases. The experimental setup they devised reflected this theoretical understanding. The stationary phase, typically a liquid, was immobilized on a solid support such as silica gel or cellulose. The mobile phase, another liquid that was immiscible with the stationary phase, was then passed through the stationary phase. The sample mixture, introduced at the beginning of the process, would then undergo separation. Compounds exhibiting a greater affinity for the mobile phase would move through the column more rapidly, while those with a stronger preference for the stationary phase would move more slowly. The separation process, therefore, was a dynamic interplay of solubility and affinity, a dance of molecular interactions within the confines of the chromatographic system. Several technical challenges demanded careful consideration. Achieving efficient and reproducible separations required optimizing several parameters. The choice of the stationary phase, the mobile phase, and the solid support all significantly impacted the separation process. The dimensions of the column, the flow rate of the mobile phase, and the temperature of the system also needed precise control. Synge and Martin meticulously investigated these factors, refining their techniques to enhance the resolution and reproducibility of their separations. Their dedication led to a robust and versatile method for separating complex mixtures. A tangible example of their work lies in the separation of acetylated amino acids. They selected this particular application to demonstrate the practical utility of their method. By carefully selecting the appropriate stationary and mobile phases, they successfully separated a mixture of acetylated amino acids, demonstrating the potential of partition chromatography for analyzing complex biochemical mixtures. This specific application highlighted the power of the technique to isolate and identify individual components within a mixture, paving the way for more detailed biochemical investigations. They published their findings, providing detailed protocols and experimental parameters, allowing other researchers to replicate and build upon their work. The initial method, while revolutionary, had limitations. The resolution of the separations depended on the careful optimization of several parameters. The technique, as initially conceived, was somewhat time-consuming and required meticulous attention to detail. However, these limitations, rather than hindering progress, stimulated further refinements. The work of Synge and Martin served as a springboard for the development of more sophisticated chromatographic techniques. Their framework provided the foundational principles upon which future generations of scientists would build. The modern relevance of the principles established by Synge and Martin remains profound. The fundamental concept of differential partitioning, the core of their approach, underlies all forms of chromatography used today, from high-performance liquid chromatography, a technique which separates compounds based on their properties, to gas chromatography, which separates volatile compounds. The underlying principle of differential partitioning continues to drive the separation process. The specific materials and techniques have, of course, advanced significantly, but the foundational principles remain unchanged. Their legacy continues to inform and guide researchers in the development of new separation technologies and in the understanding of the fundamental processes that govern chromatographic separations. The work of Synge and Martin drew directly from previous scientific insights. They were influenced by the established knowledge of liquid-liquid extraction methods, a technique that had been used for decades to separate compounds based on their solubility in different solvents. Their understanding of intermolecular forces, including van der Waals forces, hydrogen bonding, and dipole-dipole interactions, was also fundamental to their approach. These forces, which govern the interactions between molecules, dictate the solubility of a compound in a given solvent. Their understanding of these forces allowed them to predict and manipulate the separation behavior of various compounds. The direct influence of Synge and Martin's work on others is undeniable. Their pioneering work directly inspired the development of column chromatography, a technique that allowed for the separation of large quantities of complex mixtures. Their work also influenced the development of other separation techniques, including paper chromatography, which provided a simpler and more accessible method for separating mixtures. Their influence is evident in the work of James Lovelock, a scientist who developed the electron capture detector, a highly sensitive instrument used in gas chromatography. Lovelock, inspired by their work, used chromatography in his pioneering work on atmospheric analysis. In essence, the work of Synge and Martin, with its focus on the fundamental principles of separation, provided a cornerstone for modern analytical chemistry. Their contribution, therefore, transcends a single technique. It represents a fundamental shift in the way scientists approach the separation and analysis of complex mixtures. The foundational work on chromatographic separation, so effectively initiated by Synge and Martin, paved the way for their next, equally impactful endeavor, the elucidation of amino acid sequences in proteins. This project addressed a central, perplexing problem in the nascent field of biochemistry. Scientists grappled with the intricate structures of proteins, the complex molecules that performed a vast array of functions within living organisms. The challenge resided in deciphering the order in which amino acids, the building blocks of proteins, were linked together. Understanding this sequence was crucial. It dictated a protein's three-dimensional structure and, consequently, its biological activity. Synge and Martin's solution, as with their earlier chromatographic work, was both elegant and practical. They adapted partition chromatography, the very technique they had pioneered, to separate and analyze amino acids and their constituent peptides. This approach offered a powerful means of dissecting the complex mixtures arising from the partial hydrolysis of proteins. Their methods provided a pathway to identify and quantify individual amino acids, providing a critical first step in sequence determination. A key innovation was the application of paper chromatography. This technique, a simplification of their earlier column chromatography, offered a more accessible and readily available method for separating amino acids. A small sample of the protein, broken down into its constituent parts, was spotted onto a piece of filter paper. The paper was then placed in a solvent, which slowly migrated up the paper by capillary action. Different amino acids, due to their varying solubilities and affinities for the solvent and the paper, traveled at different rates, separating into distinct spots. The positions of these spots, revealed through chemical reactions, allowed for the identification and quantification of the amino acids present in the sample. This technique, though relatively simple in its execution, provided a powerful analytical tool. It allowed scientists to begin to map the amino acid composition of proteins. Furthermore, by carefully controlling the conditions of protein hydrolysis, Synge and Martin could generate smaller peptide fragments. Analyzing these fragments using paper chromatography provided clues about the sequence of amino acids within the larger protein molecule. The impact of their work was immediate and far-reaching. It provided a fundamental methodology for studying the structure of peptides and proteins. This methodology offered a practical means to determine the amino acid composition of proteins, a crucial first step in understanding their function. The methods they developed allow biochemists to tackle the complexity of protein structure with greater precision and efficiency. Consider, for example, their analysis of the peptide gramacidin S, a cyclic decaptide with antibiotic properties. Using paper chromatography, they were able to determine the sequence of amino acids in this molecule. This work, among others, demonstrated the ability of their techniques to tackle complex biological molecules. The ability to analyze and sequence peptides, in turn, provided a foundation for understanding the structure and function of larger, more complex proteins. The intellectual environment in which Synge and Martin undertook their work was shaped by prior investigations into the nature of proteins. The work of Emil Fischer, a pioneer in peptide synthesis, provided a critical foundation. Fischer's research on synthesizing peptides, specifically his understanding of the peptide bond, provided essential context. Fischer demonstrated that amino acids could be linked together to form chains, a fundamental insight that underpinned Synge and Martin's quest to understand the order of amino acids in naturally occurring proteins. Their work essentially built upon Fischer's insights, providing the means to analyze and dissect those chains that Fischer had, in a sense, created. The influence of their work on others is readily apparent in the subsequent evolution of protein sequencing techniques. Their foundational principles directly inspired the development of automated protein sequencers. The core idea of separating and analyzing amino acids, so elegantly demonstrated by paper chromatography, was adapted and refined in these automated systems. These machines could perform the labor-intensive steps of protein sequencing with increased speed and accuracy. One of the scientists who directly benefited from and built upon Synge and Martin's methods was Frederick Sanger. Sanger, employing chromatography as a key tool, went on to develop methods for sequencing insulin. His work, which ultimately earned him 2 Nobel Prizes, stands as a testament to the lasting impact of Synge and Martin's pioneering contributions. Sanger's success shows the way in which their initial methods provided a crucial foundation for the development of advanced techniques and for the eventual complete sequencing of proteins. The modern relevance of Synge and Martin's work is undeniable. While more sophisticated techniques, such as mass spectrometry and automated sequencers, have largely supplanted paper chromatography in the laboratory, the underlying principles remain fundamentally unchanged. The idea of separating molecules based on their physical and chemical properties, the core concept of partition chromatography, continues to be a cornerstone of analytical chemistry and biochemistry. The principles of amino acid analysis and sequencing, the very methods Synge and Martin pioneered, are still taught in undergraduate and graduate courses. These principles are the foundation upon which modern techniques are built. The legacy of Synge and Martin's work extends beyond its direct applications. It represents a fundamental shift in how scientists approached the study of complex biological molecules. It provided tools and methodologies that empowered researchers to unravel the mysteries of protein structure, leading to a deeper understanding of life itself. Their contributions, rooted in the innovative application of partition chromatography, represent a pivotal moment in the history of biochemistry, one that continues to shape scientific inquiry today. The culmination of Synge and Martin's groundbreaking work arrived with the recognition of their achievements by the scientific community. In 1952, they were awarded the Nobel Prize in Chemistry for their invention of partition chromatography. The announcement reverberated throughout the scientific world, validating the significance of their pioneering methods. The prize served to officially acknowledge the transformative impact of their work on the field of biochemistry and analytical chemistry. The context of this award is crucial. Before Synge and Martin, separating and analyzing complex mixtures of amino acids and proteins was a formidable challenge. Existing techniques, such as those relying on differential precipitation or fractional crystallization, were often laborious, time-consuming, and yielded limited information. Their invention, partition chromatography, offered a fundamentally new approach, providing a far more efficient and versatile method for separating and identifying these crucial biological molecules. The Nobel Prize highlighted the paradigm shift they initiated. The impact of the award extended beyond mere recognition. It served as a powerful catalyst, accelerating the adoption and refinement of chromatographic techniques. The scientific community, already impressed by the efficacy of their methods, embraced the validation the Nobel Prize provided. The award spurred further research and development, leading to the proliferation of chromatographic methods across diverse scientific disciplines. This validation spurred further research and development, leading to the proliferation of chromatographic methods across diverse scientific disciplines. It provided a powerful incentive for other scientists to explore and extend the potential of chromatography. The scientific community's reaction was overwhelmingly positive. The prize was seen as a deserved acknowledgement of their ingenuity and the profound impact of their work. It stimulated further research and development in chromatography, inspiring scientists to explore new applications and refine existing techniques. The award brought greater visibility to their work, attracting more researchers and fostering a deeper understanding of the fundamental principles underlying their approach. The prestige of the Nobel Prize undoubtedly contributed to the rapid dissemination of their methods and their widespread adoption in laboratories worldwide. The broader implications of their work are undeniable. Partition chromatography revolutionized the study of proteins, amino acids, and other complex biological molecules. It enabled scientists to analyze the composition, structure, and function of these molecules with unprecedented precision. This, in turn, accelerated progress in various fields, including biochemistry, molecular biology, and pharmaceutical science. The ability to separate and identify amino acids, for example, was critical for understanding the structure of proteins. This understanding was a necessary precursor to the development of new drugs and therapies. Specific examples of their work's influence are abundant. Numerous scientific papers and publications cited their work, demonstrating its widespread impact. For instance, the development of automated amino acid analyzers, which streamlined the process of protein sequencing, was directly influenced by their chromatographic methods. The principles they established formed the foundation for many subsequent advancements in analytical chemistry and biochemistry. Their work laid the groundwork for techniques like gas chromatography and high-performance liquid chromatography, which are standard tools in scientific laboratories today. Their influence extended into fields like food science, environmental monitoring, and clinical diagnostics. The modern relevance of their work is significant. Although more advanced chromatographic techniques have been developed, the fundamental principles of partition chromatography, as pioneered by Synge and Martin, remain central to the field. These principles are still taught in undergraduate and graduate courses in chemistry, biochemistry, and related disciplines. The concepts of separation based on differential partitioning, the importance of stationary and mobile phases, and the factors affecting retention are all directly derived from their work. The contributions are not a relic of the past. They form the conceptual bedrock upon which modern chromatographic methods are built. The influence of earlier work on separation techniques, specifically that of Mikhail T. Sweat, must be acknowledged. T. Sweat, a Russian-Italian botanist, developed a form of chromatography in 1903 to separate plant pigments. His method, using a column of adsorbent material and a solvent to separate the pigments, laid the groundwork for the development of partition chromatography. Synge and Martin, while innovating significantly upon T. Sweat's original concept, built upon his foundational work. Their understanding of the principles of adsorption and partition was crucial for developing their own techniques. The direct influence on others stemming from Synge and Martin's Nobel Prize is readily apparent. The recognition of their work inspired numerous scientists in the field of biochemistry and analytical chemistry. Their success demonstrated the power of chromatographic techniques, encouraging researchers to explore new applications and refine existing methods. The award served as a catalyst for innovation, leading to the development of new chromatographic techniques and the improvement of existing ones. The prize acted as an inspiration for those in the field, encouraging them to build upon the foundation they had established. The award's legacy is evident in the continued development and refinement of chromatographic methods, all of which build upon the pioneering work of Synge and Martin. The techniques they developed transformed the study of proteins and amino acids, and their influence on the fields of biochemistry and analytical chemistry remains profound. Their contributions continue to shape scientific inquiry, and their legacy serves as a testament to the power of innovation and the importance of recognizing groundbreaking scientific achievements. The collaborative work of Richard Laurence Millington Synge and Archer John Porter Martin, particularly their groundbreaking development of partition chromatography, irrevocably altered the landscape of biochemical and analytical research. Their achievement, recognized by the Nobel Prize, represented a pivotal moment, a testament to the transformative potential of innovative methodologies. The impact of their work extended far beyond the initial separation of amino acids, permeating numerous scientific disciplines and establishing a framework for future advancements. The essence of their innovation resided in the creation of a simple yet remarkably effective method for separating complex mixtures. Partition chromatography, a technique employing the differential distribution of substances between two immiscible phases, offered a level of precision and efficiency previously unattainable. This approach, born from their combined expertise, revolutionized the study of proteins, amino acids, and other complex biological molecules. The precise separation of these components enabled researchers to delve deeper into their structures, functions, and interactions, unlocking a wealth of new knowledge. The immediate consequence of their work was a surge in the sophistication of analytical techniques. Scientists, armed with a more potent method for separating and analyzing complex mixtures, could address questions that had previously been inaccessible. The ability to isolate and identify specific compounds, even in minute quantities, accelerated progress in various fields. The techniques they pioneered became essential tools in biochemistry, pharmacology, and other related areas, providing the foundation for countless discoveries. The influence of Synge and Martin's work is readily apparent in the evolution of chromatography itself. Their initial methods, though groundbreaking, served as a springboard for continuous refinement and development. The principles they established were adapted and expanded upon, leading to a proliferation of chromatographic techniques, each tailored to specific applications. Gas chromatography, high-performance liquid chromatography, and other modern methods are direct descendants of their pioneering efforts. These techniques, now ubiquitous in scientific laboratories, owe a profound debt to the foundational work of Synge and Martin. The impact of partition chromatography is also evident in the advancement of pharmaceutical science. The ability to isolate and purify active pharmaceutical ingredients is crucial for drug development. Chromatographic methods, developed from the principles established by Synge and Martin, play a vital role in ensuring the purity, safety, and efficacy of medications. Their work indirectly contributed to the development of new drugs and therapies, ultimately improving human health. Furthermore, the legacy of Synge and Martin extends to the field of proteomics, the large-scale study of proteins. The methods they developed laid the groundwork for the analysis of complex protein mixtures, enabling researchers to identify, quantify, and characterize proteins within biological systems. This has been instrumental in understanding cellular processes, disease mechanisms, and the development of new diagnostic tools and therapeutic interventions. The ability to analyze proteins with greater precision and sensitivity has transformed our understanding of biology at the molecular level. The intellectual courage and innovative spirit of Synge and Martin should be acknowledged. They tackled complex scientific challenges with ingenuity and determination, devising solutions that were both elegant and practical. Their willingness to explore new approaches and to challenge conventional wisdom was crucial to their success. Their work serves as an inspiration to scientists, demonstrating the importance of creativity, perseverance, and a commitment to rigorous experimentation. Their contributions also highlight the importance of collaboration in scientific progress. The partnership between Synge and Martin, combining their unique skills and perspectives, facilitated the development of a groundbreaking technique. Their ability to work together, to share ideas, and to support each other was a significant factor in their achievement. Their collaboration underscores the power of teamwork and the benefits of diverse expertise in scientific endeavors. The broader impact of their work is evident in the evolution of scientific methodology. Their emphasis on precise measurement, rigorous experimentation, and careful data analysis set a new standard for scientific inquiry. Their approach, focused on the systematic investigation of complex phenomena, has influenced generations of scientists. Their work, a model of scientific excellence, continues to shape the way research is conducted in numerous disciplines. The enduring significance of Synge and Martin's work ultimately lies in its transformative effect on scientific understanding. Their development of partition chromatography opened up new avenues of investigation, leading to a deeper understanding of the fundamental building blocks of life. Their pioneering work in separation science provided the tools and techniques necessary to explore the complexities of biological systems, contributing to advances in biochemistry, medicine, and other fields. Their legacy, a testament to the power of innovation, continues to inspire scientists to push the boundaries of knowledge and to seek new solutions to complex scientific problems. The recognition they received, culminating in the Nobel Prize, served as a catalyst for further advancements, encouraging scientists to build upon their foundation. Their contributions remain a cornerstone of modern science, a constant reminder of the transformative potential of human ingenuity. Their work continues to shape scientific inquiry, and their legacy serves as a testament to the power of innovation and the importance of recognizing groundbreaking scientific achievements.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.